I'm really happy to be here. This is, I can honestly say, the first time I've had to give a talk in a pub before, but it's not a bad deal. So when we were trying to pick subjects to talk to you guys about, Claire kind of covered regeneration, so I thought I'd give a little bit different then and talk a little bit about how we can take more from an engineering approach and take kind of aspects of nature and use them to play with biology. And so I'm going to tell you two stories. One of them is looking at how you can take inspiration from nature, do some interesting research, and then take it and actually translate it to a company that we're trying to start up and see how the path can kind of go from maybe a curiosity in academics and bring it to the point where it might have impact in our lives. And then the second one is I'm going to talk about some work that I didn't do, but I thought that would be really interesting to you. It was a colleague of mine in one of the groups I've worked in who recently did the work, and I think it's just a really cool thing that you guys can relate to in terms of cancer nanotech. So a little bit about myself. I'm originally from the northern US, right around here, and grew up and became interested in materials. I saw a presentation on how satellites open solar panels. So they have these springs that when they get hot, they expand many, many times bigger than they actually are, and they open solar panels, and it's a really cool thing. And so I went to university to study material science and metallurgy up in even further north. And while I was there, I did things like I worked for Caterpillar for a little while, studying nitride distributions in the steel and bucket tips of the big excavating cranes and things you would never expect me to be talking about now. And I got fascinated, as a final year student, I got fascinated in optical circuits and how you can design materials that allow you to take computer chips and move them from using electrons to using light, because then you can imagine that they can go faster. And so I did some work on that during my senior thesis, and then decided to go to the place where integrated circuits and all that is really happening. So I did my PhD in material science at Stanford University, and I moved out there thinking that I was going to work in the same field. But I became interested then in seeing the capabilities of the semiconductor industry and how we can pattern materials and pattern them on the same length scales that biology happens on. And so how can we start to design materials that then interact with biology in kind of interesting ways? And then because I just like to do random things, I decided to join the Koch Institute for Integrative Cancer Research at MIT as a postdoctoral fellow, and spent several years there working on various approaches to modulating wound healing. And so I looked a lot at chronic wounds, things like diabetic ulcers, venous ulcers, and ways that we can manipulate signaling that happens within tissues to regenerate, similar to what Claire was talking about, where we can take and kind of try to direct tissues to repair themselves. And then in September, I moved from MIT to Imperial College London, and now I'm a lecturer there and work on tissue engineering and kind of nanobiotechnology and things like that. And so I have a small group right now. Anna happens to be here. She's one of my students. And so there's 3 of us right now. We're getting 3 more in the fall. And what we work on is things like how we can interface materials with cells and tissues. How can we create synthetic tissues? So how can we take and just build up a tissue from scratch? And then how can we control the function of tissues? So how can we start to tell tissues what to do? And so Anna works kind of in the interface of these 2 areas. So hopefully in a couple years, she'll be giving this talk on something much more fascinating than what I'm going to tell you about. But I thought today I'd start by telling you a story about how we interface materials with cells and tissues. And one of the motivations for this, there's lots of motivations, and to be brutally honest, it was an intellectual curiosity when I first started on the project. But if you look at one of the major reasons why drugs are pulled from the market, it's because they have unintended interactions with various parts of your body, with the big one being your heart, okay? So the single most common cause of withdrawal of drugs from the market is something called prolongation of the long QT interval, or of the QT interval. So this is an ECG, or EKG, an echocardiogram representation of a heartbeat. And what happens is your heart beats and you get this kind of characteristic pattern. But what happens when you get long QT is this elongates here between this Q and T, right? So this phase here elongates, and you can run into something where your heart starts to beat irregularly, okay? And what happens here is instead of all your cells beating in unison, they start to beat kind of randomly and not in unison, so you can imagine it's much harder to start pumping blood through your body. And this can then kind of go down a very bad pathway where your heart just stops beating and starts spasming, and you pump no blood, and then you have cardiac arrest. And so this is a very serious problem with drugs. And if you look at drugs that are associated or either definitively associated or thought to interact with your heart and cause this symptom, there's many, many, many drugs across a wide variety of different conditions that you're treating. So if you're taking any of these, there's a potential that you could have this happen, right? And so one recent example is in 2010, propoxyphene was withdrawn from the US market because it dangerously prolonged the QT interval, right? So this is exactly what we're talking about. So it still happens all the time, even these days. And so what people have said is we need to screen drugs to make sure that this doesn't happen, okay? So what's going on with this? If we take a look at the heart, it's this exquisite organism or organ that has all different cells that make up the heart. So you have things like cells that make up blood vessels, but these cardiomyocytes are the real workhorses. They're the ones that beat. All right. So if we look close at a cardiomyocyte, at the membrane of the cell, or the bag that holds the cell together, what we have is a lipid bilayer. And this is made up of molecules that have hydrophilic head groups, so they like water. And then in the core, kind of between them, they have areas of oily molecules or hydrophobic molecules that don't like water. And so they organize, they self-assemble. These are similar to soap. And they form these membranes. And then within these membranes, you get things like ion channels. And these allow passage of ions and various molecules through the membrane, because if you're dissolved in water, you find it very hard to just pass through this oily kind of membrane. Okay, so these are gateways into and out of the cell. And so some molecules that are involved in heart beating are, you might have calcium ions flow into these cells in the heart, and then potassium ions flow out. And what this does is it depolarizes and repolarizes the membrane and allows the cells to contract and beat and pump the blood in your heart. The problem with a lot of drugs is they interact with a potassium channel called a hERG channel that it causes your cells to repolarize. And so if it interacts with these channels, what happens is that it can't keep doing its normal beating, okay? And so what they recommend is for any drug that you're thinking about bringing to market or want to start using, that you screen for interactions with this ion channel, all right? Now, the best way to do this is with something called patch clamping. And patch clamping is used in a wide variety of fields. It's recommended for things like pharmaceutical screening against hERG channels. It's also used in basic electrophysiology research. But it gives you a powerful way to study and also interact with cells and ion channel activity. And so what you do is you bring a glass micropipette in contact with a cell, and you apply suction. And that pulls some of that membrane up into the cell. And then there's a couple of different things you can do. One of them is called a whole cell patch, where you actually tear a hole in the membrane. And now if you have an electrode in your pipette, it gives you intracellular access into your cell. And you can either record what the ion channels are doing, so you can tell whether or not they're behaving normally, or you can actually stimulate them. So you can send in ions yourself and cause cells to do things like send off nerve impulses or cause cells to beat. The problem with this for something like drug screening is that it's very labor intensive. So this is a very nice example of taking 4 patches, so 4 different pipettes, bringing them in contact with 4 different cells and seeing what's going on. But this, as you can imagine, this is very labor intensive. It's also hard if you're doing things like neurobiology research, patching very small processes of cells. So these are called dendrites. They integrate signals between neurons and things like that. And so what we asked ourselves is there a better way that we can interface with cells that allows us to, one, not have this hole in the membrane, because causing this hole also results in the cells dying fairly rapidly. But then also, can we do something where you don't have to do this and it's very labor intensive? They just want to, let's say, have an electrode that fuses into a membrane. Everything is spontaneous. You can think about automating it and making it a much higher throughput. So this is where I kind of started working on this. And the thought was, can we take a look at what nature does? How do you incorporate these gateways into cell membranes? And so this is a representation of what one of these ion channels looks like. And it's color-coded to have either in blue parts of the protein that are hydrophilic, they like water, or that are in yellow that are hydrophobic and don't like water, right? So they're oily. So what happens is these integrate into cell membranes, and where they touch the interior of the membrane, they display almost exclusively hydrophobic amino acids. So it's like mixing oil and oil. They like to do this. But if you look at the surfaces of the protein, where they would be in contact either with water that's in your cells or outside your cells, or in contact with these lipid head groups that are also hydrophilic, you can see that they predominantly display hydrophilic amino acids. So the question is, can we take an electrode, so if this is, let's say, a metal wire, and put it through a cell, can we design something that mimics this hydrophobic core of the membrane so that it interacts specifically with the different regions of the bilayer, right? So can we put a hydrophobic band on something that's hydrophilic and just simply interact the same way nature does, right? I mean, they've had millions of years to figure this out. Why not do the same thing? Well, this is the problem that we run into. Lipid bilayers are extremely, extremely thin, okay? So if we take this circle that makes up this curvature that goes way off the page as the average diameter of a human hair, the width of this hydrophobic region of the bilayer core is a small dot that you probably can't even see here. So it's 20,000 times thinner than the thickness of your human hairs, okay? So we're talking about taking, how do you take any material and pattern it down to such a small scale? We're talking tens of atoms thick, or if that, maybe 10 atoms thick. And where I was in Silicon Valley at the time. So the idea is, you can use potentially ways that we make computer chips. So computer chips are patterned. You have millions and millions of transistors on your computer chip. And so it's very, very high control over how we can pattern these materials. But can we do the same thing here? And kind of the thought behind it was looking at ways that you can make very fine or very thin features. And so one of them, if you think about painting, but this could be any kind of thing where you go and want to just draw a very thin line. You can draw a very thin line, but it's hard as you get at the very tip here, you can get very thin, but it's extremely, extremely difficult to pattern a line of material that's just several nanometers or several atoms in diameter. On the other hand, the thing to always keep in mind is that there's always hope, right? So if you want to do this, you can, the trick to it is that if you spray materials, right? So if you spray paint something, you can spray such a thin layer of material that you can actually still see through it. So you can get very, very thin. Let's say if you're painting a wall, so this is a Banksy, and if you look here, you can see some of the wall that you can kind of see through the dress. The exact same thing we can do with materials, all right? So we want to make a metal electrode, let's say. We can take and spray atoms of something like chromium and make up a thin layer, and then spray a different metal. In this case, we're spraying gold, and then finally spray another layer of chrome. And then we can sandblast it with different ions and etch away, revealing the thickness of this gold layer, right? So now we're not talking about how fine we can draw a line, but how thin we can actually make it. And we can make things down to just a couple of atoms thick. And so the nice thing about this then is we can take advantage of a process called self-assembly, where if you have the right conditions, materials and molecules and atoms just form into a structure that you want. And one of the unique things is that with gold, there's atoms like sulfur that really like to attach to gold, but not to something like chrome oxide or chrome. And so if we take sulfur atoms and attach them to the end of an oily molecule, so something that's hydrophobic, and put them in a solution, they will specifically assemble just on our gold band. So then we've defined the thickness of this band by how thick our gold film is, and then just let molecules do what they like to do and assemble into this hydrophobic band. And so this is one of these probes. So this is looked at in an electron microscope. And this one was designed to have a silicon post and then these 3 layers of metal. So we have chrome followed by gold. This is a 5 nanometer gold band. So it's about 10-ish atoms thick. And then another chrome layer. And it creates this band. And then what we can do is subsequently attach these hydrophobic molecules to it. And so the question is, does this actually work? Do these fuse into cells? And so I spent about just over 20% of my life figuring out how to do this. And when you're in lab late at night and you want to know whether or not these actually fuse into cells, the easiest cells to get a hold of are red blood cells. So there are some generously donated red blood cells from a fabulous graduate student that fused into. We have these probes on a cantilever up here, and we can touch the cantilever against the red blood cell and then withdraw it. And what you can see is that we actually can stretch out these cells. So the post that I showed on the last picture is out in this dark region, but we've actually fused to the membrane, and there's a tether that runs out and is stretching this cell because it's fused in just like a transmembrane protein. Okay? But if we yank hard enough, so this picture, we had this cell there for hours. It just hung out nice and tensed. If you pull it out eventually, what happens is it goes back, and you can see that it returns kind of to looking like a red blood cell. So we're not damaging the cell. We're not ripping big holes in it that causes it to fall apart. And it can go back to being happy. And there's a lot of work that went into figuring out what the different molecules, so these hydrophobic molecules, how we attach them, what the properties of those are, how thick they can be, and how all of those impact how it fuses into the membrane. So we can make everything from things that fuse in very strongly to things that insert but then essentially break free. So if you want to think of something that can directly penetrate these membranes, we can design interfaces that allow us to then transit materials into cells. But I started this by saying, okay, we want to think about ways that we can make a device that will allow us to interact with cells in an automated way, in a spontaneous way. We don't need any manual intervention. And so taking this data now, what's going on is we're looking at making arrays of electrodes. So this is a fabricated device that's been fabricated out at Stanford, where we have individually addressed electrodes. In this case, I believe there's 96 of them. At the end of each electrode, we have a post, and on that post, we have a gold band. And so the idea is then you can functionalize these, have an electrode on the tip, and then interact with cells, whether they be for drug screening or for neurobiology research, looking at, once you have an electrode that's physically inside a cell, what can you learn about how it works and how it functions? So everything from looking at ion channel interactions to things like if you culture a set of neurons on this chip, how do they start to learn memory, right? How do neurons learn, and what goes on as they form neural networks? And so we formed a company recently on this, looking at developing these for various applications, such as personalized medicine and nanoscience, along with some other nanotech devices that we've made. But I thought this was a good example of how a very academic curiosity that we started with, going through and understanding how we can design something to take advantage of the guidelines that nature gives us, can be taken and then translated to, in this case, a company that hopefully will have a big impact on things like how we can screen drugs, reducing a lot of the side effects, and reducing things like unnecessary deaths because of interactions with these ion channels. All right. So that's kind of the first story that I thought we'd go over. And the next story now is by a good friend of mine who I worked in the lab with him at MIT and was interested in looking at how new ways that we can treat cancer. Okay? So everybody probably knows somebody that's had cancer. And so if we start with somebody, just an average person, the chance of them getting cancer as they age is very, very high, right? And so if we get, and let's say this is maybe lung cancer, if we look closely in it, what we see is that we get this kind of uncontrolled growth of cells. And so what happens is that these cells that are part of the tumor grow, they multiply out of control, they start to migrate, and they grow things like blood vessels. They don't do a very good job at it. They're kind of all random. They also have holes in them, so they're kind of leaky. It's kind of like doing a shoddy job on your plumbing. But they grow and eventually take over your body and cause a lot of problems. And so one of the ways that they go about doing this is they hijack a lot of the mechanisms that we use to naturally heal wounds and repair our tissues. So if we look at the commonalities between things like cancer and wound healing, what we see is that normally in something like wound healing, we have transient proliferative signaling. So our cells multiply until they don't need to multiply anymore, and then it stops. But cancer hijacks these pathways and continues to multiply. It evades things that tell it to stop growing, and it does things like grows blood vessels to provide it with nutrients and hijacks a lot of these pathways. So how does this work? So in wound healing, what happens is a bunch of molecules are released called growth factors that coordinate various processes of wound healing. They coordinate the different cells, so they do things like grow blood vessels. They take, if you have a scab, they tell the epidermis to grow over it, and eventually all of this is resolved and you form a scar. And so this is coordinated through these growth factors, and these growth factors bind on receptors in our lipid membrane and send off a cascade of signaling events. And what happens is, depending on what binds here and what happens through these pathways, it can tell the cells to do a lot of different things. So it can tell them to maybe grow blood vessels or start proliferating, but it can also do things that are bad, like tell it not to die, right? So apoptosis is where cells die. And so it can actively tell them, if you're stressed, if something's wrong, don't actually die, just keep doing what you're doing, right? And so cancer hijacks a lot of these pathways to keep itself resistant to dying and proliferating and then populating our body. And so a lot of drugs and cancer drugs have gone into blocking various aspects of these signaling pathways. So you can get mutations in these pathways that activate them. So if you have an activating mutation somewhere down here, you don't actually need the growth factor to keep signaling. Or you might overexpress these or have a mutation somewhere here. And so there's these, in bold, we have different drugs that have been developed that target different aspects of how this cell signaling goes on that tells cancer cells to do various things. And so what I'm going to point out here is erlotinib, which is something called an EGFR inhibitor. And so there's a growth factor called EGF, and it binds to its receptor. And erlotinib goes in and interacts with this complex that forms and prevents EGFR from signaling. Okay? The other class of common class of kind of cancer drugs and many chemotherapeutics instead take the approach of damaging DNA. And so if you damage DNA, it's a signal to the cell that it wants to die, right? So there's been a problem, let's kill ourselves. But cancer a lot of times evades that. And so drugs like doxorubicin go in, and when you have breaks in your DNA, tell it stabilizes it and doesn't keep it from reforming and fixing itself. And so in cells that proliferate a lot, remember, cancer likes to proliferate and keep growing. And so in cells that proliferate a lot, you can introduce more and more of these breaks and increase the DNA damage. This is also why if you try to kill cancer, you kill off things like your hair and you get ulcers in your mouth and areas of your body that have rapidly proliferating cells. So any proliferating cell in your body is susceptible to drugs that target things like cell proliferation. So these are very potent drugs that have very nasty side effects, but they generally can do quite well in some cases at attacking cancer. And so a couple years ago in the Yaffe lab at MIT, they did an interesting study where they took these two drugs, erlotinib and doxorubicin. So one inhibits growth factor signaling and EGFR signaling, and one damages DNA. And in a couple types of cancer, one's called triple-negative breast cancer. It's a kind of specific form of breast cancer that's very hard to treat. And in lung cancer, the drugs by themselves don't work very well. Okay, so what we're looking at here is the number of cells or the percent of cells that die if you give them one of these drugs. So if you give them erlotinib, so this EGFR inhibitor, you get maybe about 15% of cells that die. And if you give it doxorubicin, again, you get about 15% of cells that die. If you give them, one of the things that people do in cancer therapy is you give cocktails of drugs, okay? So if you give them together, you actually don't get any improvement, all right? And then you can ask the thing, well, what happens if you give one and then the other, all right? So if you give doxorubicin, so if you damage the DNA and then inhibit signaling, there's really no improvement, right? It'd still kill about 15%. If you give erlotinib to block the signaling and then give doxorubicin, but only give it about 1 hour apart, you have no effect. But if you give it 24 hours apart, you can get up to 500% increase in the amount of cells that you're able to kill, right? So we're giving the exact same molecules. We're just staggering them by 24 hours. So it's this curious case of that biology is not static, it's dynamic, right? It likes to move around, it likes to do things. So what's going on here? What we're doing, if you give erlotinib and then wait and give doxorubicin, is that you're blocking this signaling, and this signaling goes through and changes a bunch of things within the cell that tell the cell to stay alive. So if you block that signal, and then what happens is it no longer gets that signal, it rewires itself, and it becomes more susceptible to DNA damage and then subsequently death. So if you block the signaling for long enough, so in this case 24 hours, and then give the doxorubicin, you can actually increase the efficacy of these drugs, all right? So one of the problems with this, though, is that these are very, very potent drugs. And so they have a lot of unintended side effects. So the real kind of best thing to do would be, is there a way that we can target just the tumor, deliver our drugs to it, and avoid all the other parts of our body? Because then we could deliver more of it, you could give it, you wouldn't have all the side effects that you get from having systemic drugs. And so one way we can do this is through nanotechnology, and specifically things like nanoparticles. So if we can package the drugs into very, very small particles, these are maybe 1,000 times smaller than the width of a human hair. What happens when they flow through the tumor is, remember, tumors are very bad at making blood vessels, and they make these leaky blood vessels. And you can get them to diffuse into the tumor, whereas normally there's not these holes in the rest of your body. And so they can accumulate within the tumor. And then tumors do things like they display various proteins on their surface that you can target. So you can think about if you put something on there that binds that overexpressed protein on their surface, you might get them to diffuse in there and then bind to the cell and remain there, whereas they might not normally bind very much to a different cell that's not cancerous, all right? So my colleague went through and developed this lipid nanoparticle. So this is taking lipids, the same kind of molecules that make up cell membranes, but instead making a nanoparticle, okay? So if you look here, we have these hydrophilic head groups, and then right here we have this hydrophobic bilayer core, and then in the inside we have another hydrophilic surface, and within these little packages there's water that's trapped in the middle, okay? So it's a little bag of water. And the nice thing about erlotinib and doxorubicin is hydrophilic. It likes water, right? So it will sit happily in the center of these little particles. And erlotinib is hydrophobic. So it prefers to sit in this hydrophobic region of the bilayer, okay? So we can actually compartmentalize these drugs, two different drugs, into one particle, all right? Then we can do things like attach, so this folate, cancer cells many times, or certain types of them, overexpress folate receptors. And so by attaching these folate molecules, we can actually target it to tumors and then attach other things. These are polymers that kind of shield these from our body so they don't recognize them. They circulate longer through our system and then allows us to hopefully have more passages through the tumor so more particles will then accumulate in the tumor, all right? So we can design things that take advantage of these aspects of cancer. And the interesting thing about these particles is that because of how they're staggered, if you look at the rate at which the drugs come out of them, erlotinib, the one that sits in the hydrophobic core, is released almost over 50% of it, over 60% of it within 24 hours, So it comes out very quickly. So if you inject this in, it goes to the tumor, a lot of the erlotinib will come out very quickly. But doxorubicin takes much, much longer to come out. So at 24 hours, not even 20% come out. So a lot more of it still has to come. So the idea is if you can deliver these to the tumor, then you'll have erlotinib come out first. It will block this signaling, rewire the cell, and then later on, as this breaks down, you'll have the doxorubicin come out, okay? And then damage the cells and result in cell death. So they ran studies on this. So these are two very nasty types of cancer. One, the triple-negative breast cancer is on this side, and a version of non-small cell lung cancer is on the left. And what you can see is that if you don't do anything, so these are fluorescent tumors, they give off light that we can measure. What you see is that over time, so this is in 21 days, the tumors rapidly grow, all right, in both cases. If we give nanoparticles that only have the doxorubicin, all right, so they don't have the erlotinib, they don't inhibit the growth factor signaling, what you see is that the tumors still grow, right? And they look pretty much like the untreated ones, right? Remember at the beginning of this, I said doxorubicin by itself is kind of a crappy drug for these types of cancer. But if you deliver the particle that has both of them, what you can see is that it actually gets smaller, right? So this is probably the best example. You look at this one, and it goes from being this big down to almost disappearing. All right, so by simply incorporating these into one particle, we can take and deliver it and have dramatic effects in terms of the rate at which we can kill off these cancer cells. So to get regression, we actually have to be killing them faster than the tumors can actually grow, right? So this is what we want to see. We want to see being able to actively shrink the size of them. And so these have this interesting capability, plus they have the added benefit of we're not giving these drugs systemically. We're not injecting free drug that then goes through and affects all the other cells in your body, right? They're packaged into a little capsule and that preferentially accumulates within the tumor. And so there's a huge field right now looking at various ways that we can improve drug delivery, improve our efficacies. And I just talked about this one liposome where we can use these lipid particles. But there's a wide variety of nanoparticles that are being developed that all have unique properties and unique capabilities that can be applied to various applications, everything from cancer to regenerative medicine to fighting infection. And so there's really a ton of work going into how we design materials on the same length scales that biology interacts and plays with, and how can we design them to take advantage of that and use them to our advantage to have more effective therapeutics, more effective drugs that have less unintended side effects. And so with that, I just want to thank everybody that worked with this, my group, and then Professor Nick Malosh at Stanford, who worked with me on the stealth probes, and then Professor Paula Hammond at MIT, whose lab is responsible for these really cool little nanoparticles that I talked about, and of course the people that pay the bills. So with that, I'd be happy to take any questions that you have.
I have one. Okay. So how do you know when the doxorubicin is going to be released? So you package it inside its lifesome. How do you trigger its release of its lifesome?
It's just slowly released over time as the lipid particle breaks down. So they go up and they're actually uptaken by the cells. So they bind the folate receptors, they're taken into the cells, and then the cell starts to break it down, and it's slowly released as they kind of break down.
And is it like a fairly controlled manner? Is it fairly predictable?
It could be better. So one of the drawback to lipid particles, so there's a doxorubicin formulation called Doxil that is a lipid particle, and it has a lot of problems in terms of its use in the clinic because the dosing is not as specific as if you were to give a free drug, because not the exact same amount is going to be incorporated in every particle. The release is going to be slightly different. So there are drawbacks, and it's not as good as we'd like. But there are other particles that have better properties in terms of that, but then there's other trade-offs. And so it's an area where everybody's looking at how do you make the perfect particle, right? And so there's work going into making it better.
I can imagine the timing to be quite tricky because you need to release both the erlotinib and the doxorubicin to be within 24 hours apart. So
that's the optimal.
Yeah. Questions? Audience?
I was wondering in the slide where you were showing the mice.
Yeah.
So that's like the result is amazing. So the tumor like stop growing and actually become smaller. What are the ways to check that this drug is not affecting other systems inside the mice body?
Yeah.
Like they're not the teeth are not falling or something like that.
Well, one is to see whether the teeth are falling out, right?
Can you check that?
So when they do these, they generally do a lot of studies. So they look at bioaccumulation. So you can tag. One of the things I didn't mention about these particles is that they tag on a fluorescent dye that you can track. So when you give them, you can actually track where these particles are going, and you can do biodistribution studies that look at what organs they're specifically accumulating in and see how long they stay in the body, and do all these studies that look at actually what the pharmacokinetics are of these particles.
I had a small question about your electrode, because I really like that idea. How easy is it to get information out of it? Like, can you do it with one cell and then put a wire on it, or I don't know what you do. What can you do?
So it's currently under development. It is when it works very well. But there's a lot of tricky engineering that go in. So the way it works, we figured out, and it works very well. But turning it into a functional device has a whole set of new challenges to address, and that is currently under development.
So, but you could. So, where are you aiming for? Are you aiming for doing it on cells, or doing it on tissue, or even whole organs?
I would say all of the above. So, I mean, if you think about one cell, there's interesting things like, let's say you're looking at one neuron, and you fuse multiple ones of them into one neuron, and you have a signal that's generated. You can start to triangulate signals within a cell on their generation. You can look at integration of signals, maybe small events that then add up to something bigger. On network level, you can start to look at, how do collections of cells begin to adapt when they're hooked up, right? So how does memory development would be one. How do they learn? Things like that if you train a network. If you start going in vivo, right? So in a tissue that's in the body, there's interesting things like, could you have an intracellular pacemaker that actually paces your heart? Could you have something that modulates the neurons in your brain if you have something like chronic depression or some other things like epilepsy and whatnot? And so there's all kinds of things that I think are very interesting to potentially look at, but there's still several significant hurdles to jump to get to those points. Yeah.
I was just wondering how expensive it is to create these nanoparticles. Something like doxorubicin would be presumably quite a cheap drug, but a lot of them more expensive. But how much money needs to go into it for you to be able to create these things that work?
These aren't horribly bad. I would say they're cheaper to make than a lot of other stuff that people want to do. Lipids are people use them for all kinds of different things. You can make them synthetically, which means that generally purities are better and costs can be better and things like that. I don't know what the price per batch or price per dose of these would be. They have some other things like we're attaching fluorophores to them and things like that would increase the cost, but in something like if you were to use it as a therapeutic in people, you wouldn't put that fluorophore on, so it would reduce the cost there, and then economies of scale and things like that. So I couldn't give you a definitive amount, but it would likely be not significantly more than the drugs by themselves.
Any other question?
Just say, following on from that, what sort of is the manufacturing process? Is it taking them from electronics rather than
For the particles or the these, I don't believe that they're manufacturing yet. So this is very new. The doxyl ones, I don't know what the exact process is, and that's part of. It's not perfect, which is one of the reasons why it has some problems with doxyl in the clinic. Yeah, I'd probably want to leave it at that. I'm not an expert there, and I don't want to say.
Something that might not actually be the case. Any questions? Well, we'll stop the Q&A session there. Please grab hold of Ben and Claire and ask them as many questions as you like. They have pints, so they'll be happy. So please, can you applaud our two fantastic speakers?
Thank you.